Exposure to phthalates during pregnancy can affect a newborn's brain development, study finds. Now, scientists may have discovered a biological pathway for how this phenomenon could occur. Researchers found that in utero exposure to phthalates is linked with altered metabolism of neurotransmitters and amino acids involved in brain maturation, according to a study published Wednesday in the journal Nature Communications. The report is the first to use untargeted metabolomics, the study of all small molecules or metabolites in a biological system, to connect a mother's phthalate exposure to a newborn's metabolites, and those metabolites to neurological development, said senior study author Dr. Donghai Liang via email. This represents an important step forward in understanding how prenatal chemical exposures shape infant development at the molecular level, added Liang, an associate professor of environmental health at Emory University's Rollins School of Public Health in Atlanta. First introduced in the 1920s, phthalates are used to make plastics softer and more flexible, primarily in polyvinyl chloride, PVC products such as vinyl flooring, medical devices, children's toys, food packaging, or shower curtains. The chemicals also help lubricate substances and carry fragrances in various personal care products including deodorant, nail polish, perfumes, hair gels, sprays or shampoos, soaps, and body lotions. Phthalates are also endocrine disruptors that have been linked to preterm birth, infant genital abnormalities, childhood obesity, asthma, cancer, cardiovascular issues, and low sperm count and testosterone in men. We conducted this study because phthalates are everywhere in our daily lives, Liang said, hence their nickname everywhere chemicals, harming infant health. The findings are based on mother-newborn pairs enrolled in the Atlanta African American maternal child cohort between 2016 and 2018. In urine samples collected from 216 mothers between 8 weeks and 14 weeks of gestation at Visit 1 and 145 participants between 24 and 30 weeks gestation at Visit 2, the authors measured 8 phthalate metabolites. Participants were around age 24 on average, and their levels of some phthalate metabolites were higher than the average determined by the National Health and Nutrition Examination Survey. Within a day or two of birth, the authors collected the baby's blood via a heel stick. The team found prenatal phthalate levels were associated with lower levels of tyrosine, an amino acid and precursor to the thyroid hormone thyroxine. Thyroxine was also abnormally low in those with in utero phthalate exposure, and low thyroxine has been previously associated with greater vulnerability to illness and neurodevelopmental issues in newborns, the authors said. Tyrosine is also a precursor to the neurotransmitters dopamine, norepinephrine, and epinephrine, all of which partly contribute to the body's fight-or-flight response. Low levels of these neurotransmitters can lead to various problems, including anxiety, depression, and trouble focusing. Prenatal phthalate exposure was also linked with lower levels of the essential amino acid tryptophan, which converts into 5-hydroxytryptophan, or 5-HEP, which then turns into serotonin. Both 5-HEP and serotonin were also low. Serotonin has several critical roles in the body, including mood regulation, sleep, learning, memory, digestion, and the body's response to stress. Previous research has linked low serotonin with mood disorders such as depression and anxiety, sleep problems, and digestive issues.